pay attention to the every details of the, just to make sure that everything is identical on both sides, right? So that takes a lot of effort, but that is for macroscopic world. When it comes to the molecular world, creating a symmetric element is actually very easy. You don't need to worry too much about any other things, but it is the unsymmetrical, or what we call the asymmetric objects that are non-superimposable mirror image or chiral as you know it, creating them is actually a big challenge. And one of the easy ways of getting that would be if you have a symmetrical object to begin with and then you break the symmetry. That will be much easier to form compared to start from something else and then slowly build up to that one, right? So this is something that I will talk about. So let me just explain you the concept. So this is a prochiral molecule where we have two identical functional groups. As a result, there is a plane of symmetry here, and this center is not yet a chiral center. This is the prochiral center. But if we convert one of these two functional group to a different functional group, then this center, which was a prochiral center, will become a chiral center. Now this is of course a prochiral compound. The same thing is also true for the meso compounds, where you can have multiple chiral centers overall, but the final molecule is not chiral because there is a plane of symmetry that is present. And you can do the same maneuver in this case to convert the meso compound to a chiral compound, and this type of reactions are called the enantio group selective transformations or enantio group differentiating reactions, because these two functional groups, they are actually enantiotopic, and when you convert one of them selectively to the other one, then you actually break the symmetry. And then of course, depending on whether you convert the group on the right side or the left side, you can actually get one or the other enantiomer. So therefore this type of reactions are known as the desymmetrization reactions, and there are a lot of advantage of this type of reactions. You can generate multiple stereogenic centers at the same time through a single operation. Most importantly, you can create a stereo center that is remote from the reaction site, because most of the reactions that, or most of the enantioselective transformation that we often come across, there you create a stereo center at the reaction site but not away from that. But in this case, we can do a reaction at one point, but it will create a stereo center that is away from that. So I will give you some of the examples of this type of reactions. So our topic today will be chiral arenes. Now arenes, or the aromatic compounds, as you know, they are flat. Like if you keep on adding them, graphene is a good example of that. I think graphene got Nobel Prize back in 2009, I believe. And if you, as you know, that if you just draw a line with a pencil, put a scotch tape and take it out, that will be a layer of graphene. And if you just put together something like a 5 million layers of graphene, that will be 1 millimeter thick. So the graphene is normally considered as a one-dimensional object, right? So they are planar. Essentially, what I'm trying to get to that, the arenes are planar. So what I mean by a chiral arenes, right? Normally, if you are thinking about chiral arenes, or if you read it in the textbooks, you will come up with this type of molecules that are called axially chiral arenes, planar chiral arenes, or helically chiral arenes. They are the most common one. And there are many examples also in nature, like this cylinder cyclophane or the vancomycin, which is the first line of antibiotics. So they are also having a lot of this type of chiral arenes. Like this is an axially chiral arene, this is the biaryl ethers, and so on. So they are things. But I will talk today, I will begin with a centrally chiral arene. So what I mean by the centrally chiral arene is the following. There are two different scenarios. One case, you have an arene ring and there is a stereo center that can be right next to the arene or somewhere farther from the arene. But these are the most common examples of that, and they are very easy to prepare. There are hundreds and thousands of examples of that, but important part is that the stereogenicity, that means the chiral center, does not depend on what is present on the arene ring, right? Even though the arene ring is not there, the chiral center may still be there in many of the cases. So therefore they are most common and well explored. But what I will talk today about is this type of compounds. So now this is something we call a centrally chiral compounds, because in this case, whether the compound is chiral or not, that will depend on the nature and the position of this R1 and R2, these two groups. If they both are identical, that is R1 and R2 are identical, then this will be a meso compound. You will have a plane of symmetry here. But if they are not same, then you have a chiral compound. But the presence of both R1 and R2 is required. If both are H, that means they are identical, you will have a meso compound. Now if you exchange the position of R1 and R2, what you get is a mirror image. So that means these two are actually enantiomer. So that means how you decorate the arene, that will decide whether you will get one enantiomer or the other one. And this type of compounds are much less common, and their synthesis is quite challenging, and they are rarely studied. So that's what we thought of taking this topic for our research. So what our idea was that we want to make this type of compounds. I put only one because the other one is naturally H, so they are not the same, and we need to make that. There are two ways you can think about making through the symmetry breaking operation. We can start with the mesoarene and selectively put this R group either at this position or at this position. That will give you the two different enantiomers. And there are of course a lot of reactions of arene where you can introduce a functional group, like a Friedel-Crafts reaction is the most common one, but enantioselective Friedel-Crafts reaction. So in this case, the enantioselectivity also means site selectivity, whether you are inserting at this site or this site. Of course, nowadays there are a lot of activities over the past 15 years or 10-15 years about site activation reactions of different kinds, and it is this extremely powerful method had shortened the synthesis several fold. Like some of the reactions that, or some of the synthetic routes that used to take 10 to 15 steps now can be made in two or three steps through this process. But even then, for making this type of symmetry breaking site activation is not something that is very common. And most importantly, for having this site activation methods, you need some functional group which can act as a directing group. And as you can see, we will see afterwards, we will be creating arenes which does not have any functional group. Now the other possibility of course would be, so in this case these two are enantiotopic, as I said, these two sites, so therefore the site selective addition would be required, and this is extremely challenging. What we thought, instead we can actually construct the arene ring itself instead of having the starting with the arene ring and functionalizing it. So that will be much easier, because now we are talking about a meso substrate of this part, and then we have an acyclic compound, and we will form this reaction here, or we will form the ring here. That's what we call the enantioselective de novo arene construction. That means starting from something simple, we will actually, or starting from scratch, we will form the arene ring. So this is what we will be talking about, because this is extremely modular. Depending on what functionalities you have, you can decorate the arene ring. You can do many other things. I will show you some of the examples now on. And of course this is rarely studied. That's what we wanted to do. So this is our idea. We start with a simple meso compounds, that is indione, that is alpha beta unsaturated carbonyl, but having a carbonyl on both sides. And then we will have the other partner, that will be an alpha beta unsaturated aldehyde, or often we call enal. And you can have different decoration depending on what's your final target is. And this is the catalytic hypothesis. We will start with the secondary amine catalyst that will condense with the aldehyde to form an iminium, and this proton will be lost. What you will get is a dienamine, and of course this secondary amine is chiral. Now we will have a 4+2 cycloaddition, that's a Diels-Alder reaction through this type of an endo transition state, and that will lead to the formation of this so-called little bit of a complex Diels-Alder adduct. The catalyst is still there. Now the catalyst has to go out of this. That can happen because this proton is acidic. It can actually kick out this catalyst and regenerate the catalyst to form a double bond here. Finally, this one can undergo spontaneous aerobic oxidation to form the arene ring. Now depending on whether you want R1, R2 or R3 at which position, you can start with the enal accordingly, and you can get the product. Now I will come back to the selectivity afterwards, because in that case I mentioned that these two positions are enantiotopic, and then depending on how the regioselectivity is achieved with the catalyst, you can get different enantiomers. And overall it's a desymmetrization reaction. So now this program started in our lab back in 2013, but we were not fast enough, so there are a couple of reports appeared in between, but luckily they were not enantioselective, so we still had the opportunity to go ahead and explore the possibilities there. There was also, while we were working on this, there was another report. There was this N-heterocyclic carbene catalyst, but in both these cases they actually prepare phenol, not an unfunctionalized arene. We wanted to make an unfunctionalized arene, because having a phenol, again again, it's a functional group. You want to remove the OH, it's a bit of a work again, so we thought we'll go for the unfunctionalized one. So this is the reaction that we have developed. So we start with the meso compound here, and we have an alpha beta unsaturated carbonyl, or aldehyde more specifically. We use this proline derived secondary amine catalyst, and that gives the product with very good yield and very high enantioselectivity. So the feature is that this is very simple. You can do it in an open flash reaction. This is an open air reaction. You need in fact air for the final oxidation step, and this is quite modular. I will show you afterwards. So this is some of the scope of these reactions. Now we are changing this R group. You can have different arenes, you can have different heteroarenes, alkenes, and even alkyl group here, and all of them can be used in this case. And here are some of the other examples, like in this place we have a CH2, now we have other substituents here, and that gives the product with high enantioselectivity. Now we are thinking of how can we decorate the ring with other functional groups. So now earlier we had only one substituent at this position, now we have two here, but the problem is we get very small amount of this compound and large amount of this so-called dearomatized compound. So we need to aromatize them further. If we treat with the oxidant like a DDQ in the presence of dichloromethane, we can actually convert this mixture to a single compound. That is possible. And now this is the decoration of the other ring. This is another 6-membered ring here, and starting from this one we can actually put it. We can have two other acyclic species here. This is something similar. This is actually interesting. So we have this, we are starting with this so-called tiglic aldehyde, this dimethyl. We put the methyl at this position. We have this dimethyl here, we put the methyl in this position. Now these two are actually enantiomeric, and we are using the same catalyst. Just by changing the substrate we can get both the mirror image form of this molecule. We found some sort of a surprise when we use citral as a substrate in this case. So we were expecting that we will get only this one, but instead we also got a little bit of this compound, and this is somewhat surprising because if you look at it, this molecule, you can form two different types of dienamine, and this one is giving this product what is we are observing. This is another one what we are expecting, but that does not form. Instead, actually after the initial cycloaddition reaction, this isoprene goes out and you form this simple methylated compound, so which is somewhat of a surprising for us, and this is something that kind of bothered us, that we need to solve this problem, particularly with an unsymmetrical enal alpha beta unsaturated aldehyde. But we thought we will take care of this later. But currently from this meso compound, can you think of some of the other compound, like we thought of this Diels-Alder adduct, and we can do that very easily. We can optimize fairly quickly and you can get even better enantioselectivity in this case. There are some of the examples. You can see what are the different type of compounds you can actually use. Of course this part we can vary again. I am not showing that. It will be very similar to the one that we have seen previously. Now you can do some cool transformation. This is our product. If you just start with silica gel in open air, you actually first enolize them to form a quinol, and that undergoes oxidation to form this type of quinone. We can do that under basic condition in the presence of Cummings reagent to form this bis triplet, and then you can cut those bis triplet to form this type of chiral anthracene derivatives, which is, we can also call them centrally chiral arenes. Now what is the mechanism of this reaction? So now this is something that is a textbook chemistry. As we know, the Diels-Alder reaction proceeds via the endo transition state, right? So therefore the two right products should be, or to the right transition state should be the favorable one, energetically at least in these two cases. Now this is, I have shown example with the cyclopropyl one. You can put the cyclopropane fused chain away from the catalyst or towards the catalyst. Of course if you put it towards the catalyst, it will face some sort of a steric interaction with the other component, so therefore this will be sterically disfavored. But the two extra transition state would be both energetically disfavored and in one case sterically disfavored. So that was our hypothesis. That's what we drew in paper, but we had to provide some evidence for that. So my colleague Garima Jindel's student Mahesh Hararia, he did some DFT calculations, and he found of this for this Diels-Alder step, and he found that indeed this transition state 1, that is sterically and energetically favored, has the lowest energy, and then the other endo transition state has somewhat higher higher energy, and these two are XO1 are much higher energy, as exactly what we expected. So, and he in fact predicted the enantioselectivity to be about 98:2 enantiomeric ratio. What we get is 97:3, which is very good agreement with what we observed experimentally. Now, having done that, we are thinking that can we now extend this concept to the axially chiral molecules, and in this context we actually came to this type of CN axial chirality. Of course, this itself is not a chiral compound because you have a plane of symmetry, and although there is a rotation around, this is normally possible, but a bulky group at the ortho position of this arene can restrict the rotation, and this sigma plane becomes much more prominent now because these two rings must be orthogonal to each other. You can actually break this sigma plane by a conjugate addition of a nucleophile at this position here, and it can add to either this site or you could throw a cycloaddition reaction, but in all these cases you are generating both central chirality and axial chirality, and they are well explored, both of them. But we didn't like generating another stereo center here. As I was talking about remote stereo control, we thought of doing the reaction at this position, but the controlling the stereochemistry of the CN axis. So effectively our idea was something like this, that this double bond here should remain intact and we should be able to create this type of CN axial chirality. So this was of course unknown, that's why we took the challenge, and again the idea was to do this type of Diels-Fowler reaction that we have seen previously under the same type of catalysis, and depending on the selectivity of this Diels-Fowler reaction you will get two different enantiomers. So what is required is a regioselective 4+2 cycloaddition reaction. So once again, this is our hypothesis for the endo transition state, and in this case this maleimide ring and the arene ring, they will be perpendicular to each other, and this bulky substituent at the ortho position that should be staying away from this catalyst or the dienamine part of the reacting component, and that should give the desired level of selectivity. This is a similar report as I told earlier, but this is an achiral arenes, and so this is what we started with. If we have this R group is H, you see there is no reaction. This is not sufficiently reactive. So we tried to make it. You all know that 4+2 cycloaddition or the Diels-Fowler reaction works better if you have a electron deficient dienophile. So we put a nitro group at this position, and you can see with the nitro we started getting the desired product. It's a very small 10% yield, and the non-oxidized one or the dearomatized one we get in 75% yield. The selectivity is also not very good. So again you can actually oxidize this dearomatized one in the presence of air and pyroladine, or oxygen also you can use. Eventually we optimized using a different catalyst. Instead of diphenyl we have dimecetyl catalyst, metaxylyl catalyst, and in this case we can get the product with 97:3 enantiomeric ratio, and we are creating. So now the reaction is happening. So these two red bonds are where the bond formation is taking place, but we are controlling the stereochemistry that is at least 3 bonds away from the reaction center. So this is what we call the remote stereo control, and the scope is again quite broad depending on this on this part. This part we cannot change much, but this part we can have aromatic, heteroaromatic, alkene, alkyl, and so on. But the important thing is that if we remove this nitro group from here, you can see the reaction takes 12 days for 11% yield. So that means less than 1% yield per day. So which is of course a limitation of this method. That means we have to have the nitro group. So we thought, can we remove the nitro group from here? We can do that. So first we can reduce it to the corresponding aniline derivative, and first initially we protected that to isolate that one. We can also do the diazotization, replace the amine with an iodo, and finally when treated with H3PO3, this is something again you have studied the diazonium salt, how to convert it to the arene. So this is exactly what we do. But in all these cases we can see there is you started with about 95% EE and we are reduced a little bit. So therefore the question comes, is the CN rotational barrier not sufficient to give this stability. So to check that, we thought of heating this compound at very different temperatures, starting from room temperature up to 60 degree or 50 degree. There is no erosion, it remains the same, and from 60 onwards you can see the enantioselective excess is changing and it is finally becoming racemic. So that means at higher temperature the rotation around this CN bond is possible. So we wanted to check what is the rotational barrier. For that we use this Dennis-Courant's method. I'll not go into the detail now, and we can get the rotational barrier, which is about 30 kilocalorie per mole. Just to give you an idea what this rotational barrier mean, if you think of the racemization, that means is going to the rapid rotation. At room temperature it will take 18.4 years for this compound to racemize completely, but if you heat it at 70 degrees Celsius it takes only 7.5 days. So therefore at higher temperature it is possible, and in this case we are doing the reduction at higher temperature, so that is possibly the reaction step where the racemization is taking place. Alright, so now what about the application? So we have now developed the reaction. We can see that we can prepare the centrally chiral molecules, axially chiral molecules, and now we are exploring currently in the lab planar chiral as well as helically chiral compounds, and they are quite successful. I'm not showing those examples today, but they are possible. So now we have to look for some applications, and when we are thinking of the application of this target or this method, we actually came to this type of natural product, and this is a naturally occurring compound, first time isolated back in 2002 from this bacteria that is called anaerobic ammonium oxidation bacteria. These bacteria are found normally at the bottom of the sea, but sometimes there are some bacteria that are present in the wastewater treatment plant, like sometimes like whatever the waste water goes out from our drain, they are actually treated with some process, and there are some bacteria grows, they actually, some of these molecules are also found in those bacteria. But this originally wherever it is isolated from, that is called anaerobic ammonium oxidation bacteria. So the name comes from this reaction. So this bacteria, they convert ammonium nitrite to molecular nitrogen at the bottom of the ocean, and at the bottom of the ocean there is no light, there is no air. So that's why it is called anaerobic ammonium oxidation bacteria, and this process contributes to about 50% of the nitrogen that is found in the ocean, or that is produced in the ocean. Of course, this reaction does not happen in the open ocean. It happens inside the bacterial cell wall and in a compartment that is called anamoxosome. And in this process, of course, you generate a byproduct. So initial hypothesis that, and this green line whatever I have shown, that is the membrane from which this type of phospholipids were isolated first. So what the idea, there was a question obviously that why nature make this type of strange molecules that you can see there are multiple cyclobutane rings that are fused with each other. This is actually very difficult to make in the lab itself. Why nature is making that? What is the special purpose for that? So the initial hypothesis was that one of the intermediate is the hydrazine. Hydrazine is toxic to cells. So the initial hypothesis was that this membrane is stopping the diffusion of this hydrazine across the barrier and going to the other components of the cell. But in 2018 there was a report from Noah Burns and Stephen Boxer group. What they showed that this membrane is no different from the other type of lipid membranes in terms of diffusing or stopping the diffusion of hydrazine, but it is very special that it stops the diffusion of proton or hydroxide across the barrier, which kind of sounds strange because hydrazine is much bigger than proton, right? But you have to understand that in biological system or otherwise, proton does not travel as free proton. Proton is always solvated, and particularly in the cell with a lot of water molecules. So what you have is a protonated water cluster, and that's actually much bigger compared to hydrazine. So therefore the cell, this membrane actually prevents that diffusion, and that is very important because you might have noticed here that inside this anamoxosome the pH is 6.3, outside in the riboplasm it is 7.3. So there is a pH gradient went across this barrier, and that actually caused something called a proton motive force, and that drives the ATP synthesis within these molecules. So it is important, so because during the reaction actually this proton pump takes place from the riboplasm to the anamoxosome, and these bacteria, as they grow extremely slowly, the cell division happens once in every 2 weeks. So therefore the bacteria must maintain the pH barrier across this membrane, and this lipids helps in doing that. Okay, so that was the history of this type of molecules. Now this is a bilayer from which this type of anamoxosomes are, or this type of ladderin phospholipids are isolated. There are two examples here, it's called 5,3-PC and 3,3-PC. PC is called phosphatidylcholine, so that is this part of this molecule, and these are the two components. One is called 5-ladderanoic acid and 3-ladderinol. These are of course chiral compounds, and so our lab many years ago developed this another desymmetrization reactions, so where we again desymmetrize this type of indions, where we replace this CH bond with a C alkyl bond, and we used an unusual alkylating source, that is the nitroalkane in this case, and this is the initial mechanistic proposal that we had. Initially this nitroalkane, which is acidic at this position, that gets enolized and does a Michael addition here, again selectively between these two positions, and that is helped by this catalyst, whatever we are using, a small organic molecule, we call it bifunctional catalyst. So that was the initial idea, and then afterwards we did a thorough mechanistic studies, and we found out that this is the what we thought to be the enantiomer determining step is actually not. It is the elimination of the nitrous acid that's the enantiomer determining step. Anyway, so we eventually applied this reaction for the synthesis of 3-ladderinol and more recently 5-ladderanoic acid by using this alkylation reaction. So in case of the 3-ladderinol, we form this CH, we replace the CH with the C alkyl bond and then reduce the functional group to get to the 3-ladderinol. In this case it was a little bit more complicated because the 6-member ring was not present. So after the initial alkylation reaction, we actually had to delete this CO from this molecule, and that took quite a bit of a steps, and then eventually we get to this 5-ladderanoic acid. Alright, so now the whole point I am telling all this, the purpose that nature makes this compound was to give a sufficient density to this membrane so that the proton diffusion across the membrane does not happen, right? So now, and then there are some theoretical studies like this particular one by Peter Schreiner, and they propose that this membrane they pack in a way that there is a strong van der Waals interaction or the dispersing interaction that actually makes the membrane really dense. So we thought, can we actually make something artificial for these compounds, that instead of a simple 6-member ring here with a single bond here, we put an aromatic ring at this position, because we know that aromatic compounds, they also undergo pi stacking interaction and they actually pack very nicely. So we thought if we make this compound, whether there will be a much denser membrane, and that can stop the proton diffusion even further. So to find out how to do that, we have to make this compound. Again, this is something we have to start with. So we thought, can we do this type of an arene ring construction that I already talked about, and this is what is required, the enantioselective binary arene construction. So the first question was that, can this type of molecules be desymmetrized, or the symmetry present in this molecule can be broken using that. So this is the first substrate that we used, and I talked about this type of endo transition state that worked well. This is the second type of compound we studied that also worked pretty well in this case. Even CN axial chirality we can actually maintain quite easily through this similar type of transition state. And if you have, instead of a 3-member, you have a 4-member ring here, that should actually also work, right? In fact, it actually worked. We can desymmetrize it very easily. You can get the product with very high selectivity. Different types of the ring here, 6-membered, 5-membered, another with the tetrahydrofuran ring. We can do simple cyclobutane, also quite works very well. You can put different substituents here and so on. This is not surprising because you have the at this junction you have substituent on both sides, so therefore the selectivity is not good. And so, and this is particularly interesting because this is exactly what we are looking at in this natural product, that is this tetracyclic mesocyclohexadienone ring, hexane ion ring. So first we have to figure out how to make this compound. So we started with a very simple reaction in this case, that is the 2+2 cycloaddition reaction between malic anhydride and ethylene under photochemical condition. And Sion did this reaction, my student who graduated recently, he did this reaction in 240 gram scale, and to get about 270 gram of this final compound. This took a long time. You might have noticed the reaction time is actually 2 months, because we cannot do the irradiation continuously, but it works quite well, and you can reduce this one to get the diol that was required. Of course, if somebody is rich, they can buy these compounds. They are commercially available, but so you can see the price and you can figure out that what will be the cost of 270 gram compound if at this price. Anyway, so we protected the OH with a mesylate here. We treated this sodium sulfide to form this tetrahydrothiophene derivative, bicyclic one, then oxidized to the corresponding sulfoxide, and did the alpha chlorination with sulfuryl chloride. So now this substrate is quite important, because what we do is called Rambach-Backlund reaction, and the sulfur dioxide goes out, you deprotonate here, you kick out the chlorine, you will form these 2 rings here with the sulfoxide bridge system, and then SO goes out in this case, and you get this compound. So it took about 6 months for us to prepare this compound. It is extremely volatile, it is very difficult to purify, so we actually go ahead with we went ahead with the unpurified one, and we do another 2+2 cycloaddition. So this is the first 4-membered ring, this is the second 4-membered ring, and now we have the third 4-membered ring here, and this on oxidation give the final compound. And so once we have that, we are all set for the final reaction. But the only problem is that we have this type of unsymmetrical enal I talked about earlier, that did not give us the desired product with the good level of selectivity. In fact, when we tried this reaction, we actually got a mixture of these 2 compounds. This is the desired compound, this is the undesired one, because we have 2 different gamma protons at these positions. So both of them can actually equally react, so therefore you have almost 50-50 ratio of these 2 products, and this comes from these 2 different dienamine and derived from these 2. So we need to get only this one selectively. How do we do that? So that was a challenge that we had, and this is the overall catalytic reaction that I already talked about. And if we look at carefully, we found there are multiple problems with this catalytic cycle and this strategy, in fact. So first thing is that we have the possibility of the formation of 2 different regioisomeric dienamine that gives 2 different products. So this is something we did not want. We also knew that this cycloaddition reaction does not give very high selectivity with the ortho-aryl substituted product with an enal at this position. There are also some of the complications that this elimination sometimes it is not selective, and it can undergo oxidation before the elimination, and that gives rise to many other products. And then finally this is slow, the final oxidation step. So we thought we need to solve this problem, and our strategy was very simple. To instead of a simple alpha beta unsaturated aldehyde, you put an alkoxy group at this gamma position, or the delta position in this case, gamma in this case, and the delta will be in the dienamine. Now this is how the reaction happens. So initially the amine and aldehyde undergo condensation to form the iminium. Now this proton has to undergo elimination to form the dienamine, and because it is next to the RO group, because of the electron withdrawing inductive effect, this proton becomes more acidic compared to this one. So therefore this reaction should be much faster compared to this one. And now in this diene, which is already electron rich, has another alkoxy group at the delta position, so therefore this will have the higher homo energy compared to this one. So therefore the Diels-Alder reaction here will be much faster compared to the one at the bottom. So that is all working in our favor. And then finally, so this is basically the classical dienamine pathway. Finally here, the final aromatization step requires an aerobic oxidation reaction, and that is something that is much slower. In this case, we just need to eliminate an alcohol to get to the aromatic compound, which should be much faster. So now to find out the or to validity of this, to establish the validity of this alkoxy directed dienamine catalysis. So we thought like all of them, this actually can solve all these problems. So now to check the validity, we have two reactions started from the same substrate. One case we have the alkoxy substituted enal, other case non-alkoxy enal, and we use this catalyst to begin with, and we see whether we can actually solve all these problems. So one is the reactivity problem, as I told you, that without alkoxy the reaction takes 36 hours or 72 hours, but in this case it takes about 1 hour or 2 hours even at minus 20 degrees Celsius. Similarly, as I said earlier, that if we have the ortho-aryl substituted enal, the selectivity is not good. It's about 86:14, and now with the alkoxy one, we can actually go to 96.5. You can reduce the temperature further because the reaction takes only 4 hours instead of 72 hours, and we can get very high selectivity. So enantiomer selectivity problem we could solve. Importantly, the regioselectivity problem that was important for our synthesis, earlier we were getting almost 1:1 ratio of these two products. Now with the alkoxy one, we get only one in quantitative yield with very high enantiomer selectivity. So that's the regioselectivity issues we have. And then of course the competing pathways that I was talking about, I didn't go into the details. So in this case, without alkoxy, you can get some sort of products here with a very low yield, and now in this case we can get a single product with a very good selectivity. And then like we can go on about this other elimination reaction that I talked about that does not happen with citral in this case, and we can get only one product. So now having done that, we are all set for the desired Diels-Walder reaction. Now you can do that in this alkoxy, you can get the product with very high yield and high enantiomer selectivity. All is now required to remove this two ketone group. We can do that with B3 and triethylsilane. So effectively in just 2 steps from this, we can get to this compound. Now that we have this compound, we looked at the structure. They look very different, which is somewhat disappointing or surprising for us, but we thought, okay, let's go ahead and see how their activity is. And for that, we actually collaborated with Professor Pradeep Tarabdar from IISER Kolkata, and Subroto, his student, he did all these studies. So they actually prepared a unilamellar vesicle using a cholesterol-based lipid, and then they made the vesicle somewhat leaky using a polymer, and they had pH 7 solution inside of this HPTS. This is a dye which is a pH sensor, and they had sodium hydroxide outside. So they wanted to see if the proton is coming out or the sodium hydroxide is actually getting inside the vesicle. And this is the result. What we found that this is the top one, the purple one, is just a control experiment, and where it gives up to 78% proton leakage. This is the natural 3-laterinol, it gives up to 65%, and this is our designed compound, and that gives around 28% proton leakage. So that means it is actually providing a much denser membrane compared to the natural membrane that is formed by the bacteria. Alright, so this is the summary of that. So today I basically talked about how we synthesize this type of arenes, how we can apply them for the synthesis of centrally chiral arenes, axially chiral arenes, how we design this type of unnatural benzolaterinol, and to solve the problem existed with the previous strategy, how we developed this type of alkoxy directed dienamine catalysis and applied them for the synthesis of enantiomer selective synthesis of benzolaterine. And finally I talked a little bit about the study that we have conducted. And of course I didn't do any of this work myself. This is my present group, and this work began with Vidisha, who was a grad student back in 2013-14, and afterwards Vicky actually developed this reaction much more selectively, and then Sion took over and developed the alkoxy directed dienamine catalysis. Subhajit worked on the CN axial chirality, and then Deepak also contributed to the benzolaterinol project. So these are some of our students, and these guys are the one who are working on the exploring the other scope of this type of arene formation reaction. And our program was supported by funding agencies, and I must thank my collaborators to help us understand something that we do not have expertise of. And I thank you once again for your attention, and I'll be happy to answer any question.